So our next speaker is a longtime supporter of the ALS Association and a longtime presenter at this conference. It's Dr. Joe Beckman, PhD. He's principal investigator and Burgess and Elizabeth Jameson Chair in Health Span Research at the Linus Institute. He's the Distinguished Professor, Department of Biochemistry and Biophysics at Oregon State University. A major focus of his career work has been investigating how oxidative stress, superoxide dismutase, and zinc are involved in ALS. He's been the leader in the translational research on a copper compound, copper ATSM, as well as other innovative therapies. And I like to share this personal story because I've known Dr. Beckman for many years, is that he does not just see ALS as a interesting puzzle to figure out. He really does see the human side of this disease. The first time I met Dr. Beckman, he actually brought a person living with ALS, drove him from Corvallis to Portland for an ALS support group himself, because he felt like that would be the most useful thing to help that person. And he's always made an effort to make sure his lab students and his graduate students participate in ALS events and participate in ALS walks, because he wants to make sure they know the human side of this disease. So please join me in welcoming Dr. Joe Beckman.
Thank you. I really appreciate being invited back. And in preparing for this, I went back and started to look at what I had said 10 or 7 years ago. And it gets a little frustrating sometimes of how slow the progress is, or you'd like to go faster. So what I'd like to do today is talk about this compound, copper ATSM, which is showing some promise. And there's a lot of actually debate, or the right to try and so forth. I wanted to try to explain some of the reasons why drug development goes slow and some of the challenges with it. So it's being developed by a company called Collaborative Medicinal Developments, which is really only half a dozen people. It's a virtual pharmaceutical company. I'm a scientific advisor for them. I don't have any financial connections, and I receive no funding from them. But I like to say I'm their cheerleader in trying to say these are things that we need to move forward. I also have another company that develops mass spec, because we've actually had to invent a lot of different technologies for the work we're doing. And this mass spectrometry technology is helping us understand what's happening in ALS, but it actually has a lot of other implications. It's basically moving beyond DNA and looking at the tangles and all the other things that are going bad inside of the brain. And I have a personal connection that just started or came up with a friend who was in the military who developed ALS. So I'm going to start with this picture of an aluminum canoe in my backyard that my wife keeps wanting to give away. So I got out of the Army in 1979, and I had been in Korea. And there was this ad that was kind of amazing. You could buy a canoe for 5 boxes of Uncle Ben's rice and $150. So I got the 5 boxes, I sent it in, I bought a canoe, and I called up my friend who I had known in the Army, and that was Tom Bradshaw. And he was a West Pointer, so that made me a little nervous at first when I met him in Korea. Pretty straight-laced guy, but he was a true gentleman. And I think there's a lot to really nice people end up getting ALS. It's completely unjustified. So Tom and I put the canoe on top of a car. We took 2 weeks off, and we went canoeing throughout Maine. So I lost touch with him, actually, after a few years, and I was landing in Rome last summer, and I got an email from Tom, and I thought, this is really great. But I opened it up, and he said, I just learned I've got ALS, and I will find out that you're working on it. So it's one of those, there actually is a large community of veterans, too, that are developing the disease. So the Ice Bucket Challenge happened in 2014, and it's been about 5 years, and you may wonder about what happened with it. Why after 5 years aren't we there? Well, I've actually been working on the disease for 25 years, and I feel like I understand in some ways less about the disease than what I thought I knew a few years ago. And I just want to briefly mention, so the photo on the right is the president of OSU, Ed Ray, and he's about to have ice water dumped on his head. And the woman in the middle, Pam, Bilby did her PhD with me. And I want you to remember that because at the end, she's actually helped develop another drug that I think's got a lot of potential for treating the disease. And it's one of these things of looking at a lot of different places is really important for the disease. And I want to use this analogy last year. I've been studying swarm behavior for optimization. But it's basically the idea of, if you have flocks of fish or birds floating around and they're hunting for insects, that they go their own ways and start to search, but if one starts to have success, the other bird's kind of watching and doing its own thing, but it's seeing how's he doing. And then pretty soon they can start to swarm in, okay? And basically, swarm optimization works pretty well, and it's something that the pharmaceutical industry is really good at. Once you have a pretty good lead, you can bring a lot of people, a lot of resources to it. You find a rich area, and you can improve that. Things like the ALS money basically help you. A lot of people search out in space where you don't see anybody yet and find those first insects that are tasty to bring them in, okay? And that's one of the key things about research, and I'll come back to this. There's a lot of things that are really promising, but there's still a lot of space to search. And it's important to keep doing that searching. And you heard about collaborations, and one of the great things for me is basically I've met a lot of friends around the world. And a number of these, this is the one point in South America where you can stand at that particular spot and you can watch the sun rise and set over the Atlantic. So it's in the country of Uruguay, and the arrow points to that spot where we were standing. But Alvaro Estevez is now with me at Oregon State University, and we've been collaborating for 25 years. And Luis Barbado is also, we've been working together. And there's another drug called Mastinibid that came from Luis's work and his graduate student, now Dr. Emiliano Trias. So that collaboration with South America has been great. And the other collaboration we had is in Australia, which I really like this photograph when I was looking for an upside-down map of Australia. But you can see here the cat's head and then the dog's head. So there's a group in Melbourne that discovered copper ATSM in treating ALS. And I had a graduate student who went down to Melbourne and was working there, and he brought it back to my lab. And so this is a very important day for us that Blaine Roberts, who's in the back, and his wife Ann, and a graduate student are working with our people in the basement of the ALS building, Agricultural Life Sciences building at Oregon State. But I had to have my lab there. And that's when we realized that we really needed to try copper ATSM in a new model of ALS. So in 2012, I gave a talk here, and I was able to show that the first results that a new drug actually kept these mice alive for a year. And that was a pretty phenomenal result, which, because the SRD model of mice, which I'll explain a bit more about, is really harsh. It's really hard to treat these mice. And this is the compound, and it's copper ATSM, okay? And basically, the orange ball in the middle is an atom of copper, and it's surrounded by a very flat planar molecule that's hydrophobic, and it can carry copper into the brain, okay? And it was used as a PET imaging agent to take radioactive copper to visualize tumors. And so we knew in humans it gets into the brain immediately. Crossing the blood-brain barrier is not a problem. It happens in minutes. Well, a lot of the work that I'll talk about has led to a phase 1 trial that was done in Australia. And here's some of the results that are coming out from it. A phase 1 trial is basically just to determine safety. Is the drug safe? But the nice thing about phase 1 studies, it's usually done in healthy male volunteers, college students who will do anything for a little extra money. But in this case, you could take the drug directly to patients. And one of the things that the Australian review committee said you could do is the idea was to treat for just 28 days, but they said, well, if it's tolerated well, we want you to extend it to 6 months. So in fact, this phase 1 study actually gave a lot more data about long-term treatment. So there were 6 patients that were treated per dose, and they started at a very low dose of 3 milligrams per day. And they're at up to they actually are beyond 72 milligrams per day. But that's going to be the dose going forward. And so there are a lot of blood draws and a lot of safety. Different things were done to test the efficacy. But it's important to know that this drug actually can't tell you if the drug works, okay? It's just a very small study to see is the drug safe. And to explain what's going on here, I need to explain a little bit about copper. So each of us has about 100 milligrams of copper in our body. And we breathe in oxygen, and essentially every molecule of oxygen we breathe touches an atom of copper in order for the body to use it, okay? So it's critical. It's like an electrical conductor that's needed for us to be able to make energy and to defend against oxygen. Now, 100 milligrams is a piece of copper wire that's about that long. And the body regulates copper very carefully because if you make it twice as long, that's a lethal dose. It causes all sorts of problems to the liver and to the kidney. And to evaluate the safety of copper ATSM when you're doing 72 milligrams a day, you're giving every day about 30 milligrams of copper, okay? And what we're doing is wrapping it into a molecule that can carry it into the brain and into the liver very effectively. So when we presented this to pharmaceutical companies and to study sessions and so forth, they say, you're crazy. You're going to kill people with copper toxicity. And it's a very reasonable expectation, a very scary thing that you have to worry about. The good news is that it holds on to copper so tightly that most of it is secreted and it releases copper really to places that's needed. So it turns out the safety actually works. And now we have a dose, or as it's been identified, to move on to phase 2 studies where you randomize the drug and start to see what it can do. There we go. So last December, the first results from the phase 1 trial were talked about at a motor neuron meeting in Scotland. And it mentions here that there's some very promising studies, and this has been picked up and created a lot of enthusiasm. So this is just the rate of progression. So what happened at the 3 lowest doses is there was almost no effect on the disease progression, but at 72 milligrams, the progression is less than half for other patients. And this is still a tenth of the dose we had to give to mice to have efficacy, so it was about where you would expect to see a result. But keep in mind, that's only 6 patients, and they're not randomized, okay? So you really can't say that this is going to generalize or be a treatment. The second is that the forced vital capacity measurements actually improved. And that actually, no one's really seen that before in trials. So it's created a lot of excitement about physicians, and it's really helping accelerate the interest in moving this to phase 2 trials, that in several of the patients, their respiratory function actually increased at the higher doses. They also did a study in Parkinson's disease, and the drug is actually showing even better effects in the Parkinson's patients. So this is a functional scale where going down is doing better. So the drug is doing a little bit more than you might anticipate based on the animal studies. So in summary, what's been learned from the phase 1 studies? It's relatively safe, but there are some patients that had to withdraw from the studies. The copper ATSM is very tough to formulate, okay? So patients were having to take 24 large pills. And for patients that have difficult swallowing, taking big pills is a problem. So the other thing is that the absorption is variable. So they have to reformulate the disease, and they have to go through another phase 1 study to prove similar results are occurring. The good thing is that there's low doses have no observable effect on the disease progression. And right now, you can't say that this drug actually works, okay? Even though it looks promising, there's all sorts of statistical reasons why you can't say that this is truly working. And so now they're planning on the phase 2 trials, which is basically scaling up and starting to look for efficacy. And one of the things to keep in mind is what will it cost, okay? So, there's a joke that is, how do you make a small fortune in the pharmaceutical industry? And that is, start with a large fortune. So just to make enough drug for the phase 2 trials, I can't really talk about details, but you can put it in the range of $1 to $10 million, okay, just to treat the next 200 patients. And then when you go to the phase 2 trials to actually do the clinical work, it's in the range of $20 to $80 million, okay, depending where you do it, how many people you enroll. So it's putting a big bet on this right now as to seeing what happens. And it's understanding that there's a lot of different processes that play, a lot of different opinions that you have to come together and try to rationalize and get a lot of people to agree. And that's one of the reasons why it takes so many years to get this moving. And then really, who's going to step up and say, I'm going to commit those kinds of resources to make it happen? So I want to step back and say, I am a basic scientist, and I really have started to study this disease because of the interest. And it goes back, the first mutation that was discovered was in 1993, and it was SOD1. So it's a protein, and we heard about different genes and how the DNA is moved. I actually have studied SOD since the early 80s, actually late 70s. And Erwin Friedovich, the man on the top who discovered it, was on my PhD committee. And we were working on this for a lot of reasons. We actually turned it into a drug to treat stroke. And we discovered a side reaction that could be potentially toxic. And when the SOD mutations were uncovered, that's when we started to work on SOD and trying to understand the physical basis of it. But one of the key things here is at the center of this is a copper atom, okay? And that's the reason why copper ATSM, why we first went to investigate it, because we found that in animal models, copper was missing from a fair amount of the protein. And it's a real dilemma. We've heard that 2 to 3% of patients have mutations, but within that one gene, there's over 180 different mutations that have been discovered. And the crazy thing is, moving one carbon atom in this protein is enough to cause the protein to become toxic, okay? And yet you have a lot of this protein in every cell in your body at birth, and you can be 80 years old before the disease starts, okay? So there are a lot of puzzles to figure out about what's going on and what's causing the disease. And we've approached this in many different ways. I'm a biochemist. We produce the protein. We've studied it to death, as many other people have. We work with cultured cells, and we work with motor neurons, which you can purify. And we can study a process that causes these cells to die, and we can establish that mutant SOD will cause that problem, or it can also replicate this in a test tube. And we also work on the cells that surround motor neurons. We tend to think of the brain and the spinal cord as being neurons, but you have 10 times as many glial cells around it. And these are support cells, and these get changed. And so the yellow here is showing one process that's showing other cells that are causing damage. And it's really key because that's probably what's driving the progression of the disease. We lose motor neurons throughout life, but what causes one set of motor neurons after another to progressively die? And what we're looking for is a way to stop that progression, because then you could turn this into post-polio syndrome. And out of the 100 genes that yet need to be defined, a lot of them are going to affect that progression process. And so one of the things, there are all those mutations that have been discovered, but the amazing thing is that copper zinc SOD is the one mutation where you can take that piece of DNA from a human and you can put it in a mouse or a zebrafish or a rat or a pig or fruit flies, and they get motor neuron disease. If you do that with the other mutations, the other 50, you really don't get a very good model of motor neuron disease. And that's been one of the problems, is trying to figure out what do all these other things do that's related to motor neuron disease. It also turns out that dogs have a spontaneous mutation in SOD, and so many dog breeds are susceptible to developing a form of ALS. So it's not just humans that have this disease. And this ALS phenotype, this motor neuron degeneration that happens in mice and rats, is really robust. But the model's actually fallen out of favor. And part of it is shown here. So the mice will develop the disease. They'll look fine till about 100 days. And within 30 days, they develop the phenotype of this. So someone talked about they didn't harm mice. I'm afraid a lot of mice in my lab develop ALS. And so this was one treatment that was tried that's a common antibiotic that's thought to affect the glial cells or the microglia. And they were excited about this 10% extension of life. The NIH put $20 million into doing a clinical trial on it, and it failed. But if you look at this, the mice all died at the same age, okay? So there's $1 million prize if you could extend the life by 25%. And no one could do it for a number of years. So we were really excited when we had copper ATSM and we hit that 25% extension of life. But there was something else that we were studying. Well, I need to step back for a minute. So this SRD mouse also became kind of a poster child for the importance for rigor in science. So this is a graph of studies that were published showing a protective effect of some treatment that's in the light green, blue, green bars. And then if the same drugs were tried by the ALS therapy development in Boston, they had zero effect. That's shown in the black bars. And so that kind of contributes to this idea this model yields random results. So I mentioned that we worked closely with people in Australia, and Peter Crouch is an important collaborator. He and I did the same study at 2 different doses, and he put this graph together that showed basically as we increase doses from 2 different places, you get a linear response. I was really excited 2 years ago, maybe it's 3 now. The ALS DDI repeated the study, and they got a protective effect in a pharmaceutical company, Teva, actually repeated this at 2 doses. So multiple people can repeat the study, and that's actually never been done before in ALS. And we were actually studying something else about what makes the disease worse. And the copper ATSM, in this case, actually was extremely protective. So we had mice, this is what I presented 6 years ago. If we were able to take mice out to a year, we repeated the study in a more rigorous fashion with more animals. And the animals went out to 2 years, okay? So the curious thing is when I present this to the ALS community, they're amazed at the 25% extension of life. Nobody believes this, okay? And no one's ever repeated it yet. We have, but so it was too good of a result to be true. It actually took us 2 years to publish the result too. Now, people are saying you should only try this drug in familial SOD patients. The Australian studies are actually done in sporadic and familial patients, and the effects are protective in both. I've been working with Chris Shaw, who's up in Vancouver, Canada. He's been studying the Guam form of ALS, and he makes a very strong case that it's not that toxin BMAA, but rather there's a cholesterol molecule with a sugar attached to it that's causing the problems. It's very hydrophobic. So he's been studying this for quite a long time, and we collaborated on this experiment. It was just published last week that the copper ATSM was able to protect these mice from this toxicity. People tend not to remember, but the brain is actually people think of the liver as making cholesterol, but 25% of the synthesis of cholesterol is in the brain. So there's a whole chemistry of lipids that's really critical in the brain. Now, why would this happen? It's not an SOD model. And so there are organelles inside the body that produce energy, and copper ATSM is also doing things to protect the mitochondria. And I'll come back to that in a minute. So we've spent a lot of time of developing different derivatives of copper ATSM and testing to see how they will work and trying to find ways. But what we're excited about is this is finally moving forward into patients, and it looks like we'll find out in another year or two whether it's actually going to work generally. But I'm very excited that it's actually working in a broader range of diseases. And there will be an abstract presented at the next motor neuron meeting in Perth, Australia, that'll show it works actually on another major mutation that causes ALS. So we've had, I really set out, I'm a PhD working on pine trees that started to study ALS. But because of understanding the mechanisms, we've actually had a lot of luck of discovering new drugs that have a lot of potential. So there's copper ATSM. Mestinibid is a drug that inhibits glial proliferation. It works on mast cells. And so with Luis, we've actually managed to get enough studies that that's gone into phase 3 trials. We've done all the preclinical work for that. We think Riluzole works by a completely different mechanism that other people will do. And we think if you put it in discontinuously, give it for a few days and stop and start again, it will work better. It definitely does in mice and in cultured cells. The reviewers so far are saying that it's unethical to try it in patients, but we'll keep hammering on them. And then we've heard about Darivon or Radicava, and there's another trial using urate. That actually works on the SOD-based mechanism, so we're studying what that is. But I want to show you that we've been working on something else as well. So the first part is that when you're doing basic research, you're really hitting your head against the wall. ALS is a really tough problem with many layers. And you can see I like water themes a lot, so I like this slide of basically we're going to try things and we will wreck on the rocks fairly often. But you still need to do basic research. So this is Blossom Bar on the Rogue River, and it looks impossible to get through. But in fact, there's a way that you can go through it. But this is what you're doing with basic research, is you're exploring lots of channels, and you're going to hit a lot of rocks. You're going to fail 99% of the time. But there is a way through it when you figure out how to go to the left and go to the right and back paddle here. And that's how you're going to find a path to do what looks like it's absolutely impossible. So the last one, I want to show what we've been doing with the money that we got from the Ice Bucket Challenge that Lance arranged for us for a few years ago. So the first diagram shows a cell, and inside of it is a mitochondria, which is often called the engine. And my collaborator, Tori Hagen, was showing that there's a small molecule that gets depleted and really damages mitochondria. It's a natural antioxidant. And it's a completely different compartment in the mitochondria. So we had developed a lot of synthetic chemistry capabilities. So we were able to attach a molecule to it that would drag it through the membrane and accumulate in the mitochondria. Now, it sounds crazy to do this because it's actually bigger than anyone had done before. And getting it to work in cells would be amazing. But what we've seen in the past years, we actually gave this to a mouse model of Alzheimer's disease. And so this is showing a picture of mitochondria. So this is a healthy mouse, and these are mitochondria in an electron microscope. In this Alzheimer's model, the mitochondria become vacuolized, they blow up. And when I saw this picture, I was amazed because it's actually what you see in the SOD mouse models and some of the other models, a lot of mitochondrial damage. And just injecting that compound every other day into the belly, it actually got into the brain. And so that's a control for that. And you can see the mitochondria over here are not looking like this, but more like the controls. There's actually, you can show lots of other functional data that it's actually working. So we're really excited about this molecule and trying to get it into down this path to clinical trials. And so hopefully in 10 years we'll have it there or faster. So I'd like to stop and thank you all for your attention.